Hi all of you. So in this video we're going to learn about translation in eukaryotes. So before watching this session, please watch the video on translation in prokaryotes also, after which you can watch this session, right? Yes. So what is translation? So we have already seen the translation process is nothing but conversion of information from an mRNA to a protein, is it not? So mRNA is made of nucleotides, and these nucleotides are arranged in such a way that the first three nucleotides will make the first codon, then the nucleotides 4, 5 and 6 will make the second codon, right? And then 7, 8, 9 will make the third codon, is it not? So the mRNA is in the 5 prime 3 prime direction, and it is composed of so much of codons, and these codons, each of the codon will code for one amino acids, right? So depending on these amino acids, what amino acids the codon is coding for, the amino acids will be recruited with the help of tRNA, and these amino acids will be linked with the help of peptide bond, and a polypeptide chain is formed. Now this polypeptide chain is formed from N terminal to C terminal, is right? So this is how the translation process is going to take place, and this process takes place in the cytoplasm, and protein biosynthesis takes place in the cytoplasm, right? And what is the main structure that is involved here? The ribosomes, right? It's a ribosome. So the requirements for protein biosynthesis is nothing but mRNA and all of the standard amino acids, because in eukaryotes, if the amino acids are missing, then translation process is not going to take place, right? So if the amino acids are missing, the translation process cannot happen smoothly. So in eukaryotes, this is a very must requirement. Then tRNA, about 32 tRNAs are available to carry the amino acids from the cytoplasm, which is available in the cytoplasm, to the ribosomal translation machinery. Alright, these ribosomes, you can see them linked to the endoplasmic reticulum also. Right? In that case, you will call that endoplasmic reticulum as rough endoplasmic reticulum, right? So in the ribosomes, the protein synthesis initiation is going to take place. The protein synthesis is going to take place, and that is nothing but a ribonucleoprotein, which is composed of rRNA and protein. Now also similar to prokaryotes, even the eukaryotes will have initiation factors, elongation factors and release factors. It's very complex that you have more initiation factors, elongation factors and release factors involved in this, right? So to distinguish between the prokaryotes and eukaryotes, we write a small e in front of the IF. Okay, so IF is nothing but initiation factor, and the small e denotes for eukaryote. So eukaryotic initiation factor. Similarly, for elongation factor, we write a small e and capital E and capital F. Right, so even for release factors, it's eukaryotic RF, eukaryotic RF release factor. So this is how we denote the factors in the case of eukaryotes. Also the energy from ATP, GTP, all these are requirements in case of eukaryotic translation process. Now here, even the several ribosomes will operate together on a single mRNA, that you call it as a polyribosomal structure. So along single mRNA, when several ribosomes operate together, the process will speed up, right? So this is how the translation process will take place in case of eukaryotes, and these are the requirements. Now the basic differences we have seen this already in the session on translation and prokaryotes. So we'll just quickly refresh our memory. So prokaryotes, they synthesize all 20 amino acids on their own. Eukaryotes, we have essential, non-essential amino acids in separately, right? So all of which are necessary. So some of which we have to consume it in the diet. That is also very much necessary, because absence of these amino acids will affect the protein synthesis. And then ribosomes, in case of eukaryotes, it is, if you can see, it's 80S ribosome made of 40S and 60S, and the proteins in ribosomes, this will be about 80, and they have A and P site only. So how is it different from the eukaryotes? Prokaryotes have A, P, E sites. So it's different from prokaryotes by having only A and P site and not E site, right? And in prokaryotes, transcription and translation will take place simultaneously. But in case of eukaryotes, it's not like that, because both transcription translation are going to take place in separate organelles, right? So transcription is going to take place in the nucleus and translation is going to take place in the cytoplasm. So initially after transcription of the DNA, the product that you get will be called as the primary transcript. The primary transcript has to undergo post-transcriptional modifications, after which it will become the mature mRNA. Now this mRNA leaves the nucleus and enters the cytoplasm. In the cytoplasm, it reaches the ribosomes, wherein all of the translational factors will come together and the process will take place, right? So these are the basic differences. And few other differences. It's a prokaryotes polycistronic mRNA, that is one mRNA can code for more than one protein, whereas in eukaryotes it's a monocistronic mRNA, so it's very highly specific. One mRNA is for only one protein, right? And if you can see the speed, 18 amino acids are incorporated per second, right? So what does it mean? You have an mRNA, you have multiple codons on the mRNA, right? You have multiple codons, and for each codon there is a specific amino acid that will be brought to the ribosomal machinery, translation machinery by the tRNA, is it not? And those amino acids will get linked together by peptide bonds. So this is what is translation. So 18 amino acids are going to get incorporated per second in case of prokaryotes, whereas if you see in case of eukaryotes, about 6 amino acids per second is going to get incorporated. So it is much slower, no? Why is it slower? Because the machinery is very complex in case of eukaryotes. The translation machinery is very complex and it has high fidelity rate, right? Yes. So these are the basic differences that we had seen about the prokaryotes and eukaryotes. Now let us get into the steps of translation. So what did I tell you in the prokaryotes itself? The 3 steps of translation: initiation, elongation and termination. But even before that, we have to see one important step, that is going to be the charging of the tRNA with the amino acid. This is very important because we need to charge the tRNA with the amino acid first. So how does this happen? How does this happen? With the help of the enzyme amino acyl tRNA synthetase, and this enzyme is specific for each amino acid, and thus that functions with high accuracy because these amino acids have to be recruited corresponding to the codon, is it not? So this happens with high accuracy, and this is a this is a energy requiring process. So if you take, let's take this, look at this picture. There is the amino acid, it combines with ATP, and the enzyme, what is the enzyme? Amino acyl tRNA synthetase. Now that forms a complex amino acyl AMP enzyme complex after it releases a pyrophosphate. Yes, so this means that in this process we are going to use 2 high energy phosphates. Yes, in this process we are going to use 2 high energy phosphates. Now after this complex is formed, yes, after this complex is formed, from the complex the tRNA, yes, it reaches the tRNA. Here and where what happens, the AMP is removed, enzyme is removed, and the amino acid will sit on the tRNA, is captured by the tRNA, and the tRNA amino acyl tRNA is synthesized, right? Amino acyl tRNA is synthesized. So this kind of tRNA you call it as charged tRNA. Yes, charged tRNA, and this amino acid you call it as activated amino acid. Yes, activated amino acid. Alright, now here if you can see the charging reactions have an error rate. Yes, that is an incorrect amino acid can get esterified with the tRNA. So it has it has an error rate of less than 10 power minus 4. That is why we had seen already it happens with high accuracy, right? So to which area does this amino acid get bound to? In the 3' end of your tRNA you have the sequence. Yes, you have the sequence CCA, right? It's the amino acid acceptor arm. So to the hydroxyl group of this A, the amino acid gets linked. Yes, that is how the tRNA is being charged, and we use 2 high energy phosphates in this reaction, right? So yes, so this is how we charge the tRNA. After which, now we are going to see about the initiation, elongation and termination steps. These are the steps of translation. So again, it is all of which are same compared to the prokaryotes. Okay, in eukaryotes also it is the same, the steps are similar compared to the eukaryotes, but it's little bit compared to your prokaryotes, but it's little bit complex, right? It is little bit complex, and also the initiation factors, elongation factors all will differ, right, to some extent. So let us see how it happens. Now initiation, you have at least 10 eukaryotic initiation factor that participates, whereas in prokaryotes we have seen only 3 initiation factors that participate. So here there are about 10 eukaryotic initiation factor that participate in steps. If you can see, the first step is dissociation of ribosome. Yes, it is the first step in the, even in prokaryotes also. But the ribosomal subunit differs there in the prokaryotes and eukaryotes, so that is the difference. So first you have to dissociate the 2 subunits of the ribosome, and then you have to form a 43S pre-initiation complex, and then you form a 48S initiation complex, and then you form a 80S initiation complex. Yes, now let's see how that happens. So 80S is a ribosome. It has 2 subunits. What are the 2 subunits, all of you? It is 40S and the larger subunit is 60S, is it not? Now we are going to dissociate this. So the ribosome is going to get dissociated. So after it gets dissociated, the only the 40S ribosome we are going to deal with, right? 40S, 60S gets dissociated, and 40S only we are going to deal with. Now comes into play the eukaryotic initiation factors. Eukaryotic initiation factor 1 and eukaryotic initiation factor 3, both of which binds to the 40S. Now what is the function of these two? The function of these two is to prevent the reassociation of, yes, it is to prevent the reassociation of the 60S to the 40S. Yes, so this reassociation is prevented by the binding of this eukaryotic initiation factors to the 40S ribosome. Now after which what happens, as a next step, the eukaryotic initiation factor 2 along with GTP, yes, it is associated with the GTP. Now this will bind with the initiator tRNA. Yes, what is the initiator codon, all of you? Initiator codon is always AUG, is it not? Now this AUG will code for which amino acid? This codes for methionine. Yes, it codes for methionine. So what happens? The tRNA in the cytoplasm is charged with the methionine. Yes, it gets charged with the methionine. Now at this point I would like to remind you that in prokaryotes, how is this initiator methionine gets attached? By a formyl methionine it gets attached. The initiator methionine alone gets attached as formyl methionine, but in case of eukaryotes it's not like that. In case of eukaryotes only the methionine is attached, right? So but how do we denote this tRNA? The initiator tRNA that is carrying the methionine, yes, the tRNA that is carrying the methionine for the initiator codon will be denoted like this. Yes, will be denoted like this, tRNA methionine with a superscript I. Yes, with a superscript I. So also remember that we have 2 separate tRNAs for the methionine itself. Yes, so one tRNA for the initiator methionine is different. Yes, we have for the initiator methionine is different, and also we have a different tRNA that can carry methionine for other codons, other AUGs that is present along the length of the mRNA. So not just in the first codon you have methionine, you may have methionine, you may have AUG codons along the length of mRNA at different places also, is it not? So that, or for carrying the methionine for those codons, we have a separate tRNA, and also separate tRNA for the initiator methionine also. Yes, so this also holds good for prokaryotes also. So in eukaryotes we have separate tRNAs, so that we carry methionine for initiator codon, and we can carry methionine for the other codons along the length of the mRNA. Now let's come back. The eukaryotic initiation factor 2 along with GTP is going to bind with the tRNA that is carrying the methionine for the first codon, right? So that you, how do you express it? You express it by a superscript I. Okay, so this is how you denote it. So now this one, what happens now, it comes and attaches itself to the 40S ribosome. Okay, so this eukaryotic initiation factor 2 with GTP, we call it as a binary complex. Yes, so when this binary complex combines with this tRNA, we call it as a ternary complex. Yes, we call it as a ternary complex. Now this ternary complex comes to the 40S ribosome. Yes, comes to the 40S ribosome, to which the initiating factor 1 and 3 have bound to. So as it comes, now let us draw the structure again. As it comes, what happens? We have the eukaryotic initiation factor 1. Yes, eukaryotic initiation factor 3, and also we have eukaryotic initiation factor 2 with GTP. Yes, that has brought our tRNA. Yes, that has brought our tRNA here, that is carrying the methionine, right? That's carrying the methionine. So this is the 43S pre-initiation complex. That is the 43S pre-initiation complex. All right. Yes. So thus we have formed the 43S pre-initiation complex. Now our next step. Yes, what is going to be our next step? Our next step is to bring the mRNA to the place. Yes, our next step is to bring the mRNA to the place. So for which we need to first activate the mRNA. Yes, we need to first activate the mRNA. So after transcription, post-transcriptional modifications has happened to the mRNA. So in which the mRNA has been added, the capping has been done and tailing also has been done. Is it not? So what is the purpose of this capping? The purpose of this capping and tailing is, it is to prevent the mRNA from getting attacked by nucleases. So not just that, the capping also helps in guiding the mRNA towards your ribosomes for translation, right? So now there is one initiation, eukaryotic initiation factor 4F. Yes, you have to remember, it is eukaryotic initiation factor 4F. That is a complex that is made of 4E, 4G and 4A. Yes, that is a complex that is made of 4E, 4G and 4A. That is eukaryotic initiation factor 4F. Okay, so now this binds with the cap. Okay, this binds with the cap. Now this is again a energy requiring process. Now this binds with the cap. So let's draw the mRNA with the cap. To the cap you have the eukaryotic initiation factor 4F that is being bound, right? Also you have another protein which is poly A tail binding protein that will bind to the tail. Poly A tail binding protein, right? So together you also have other factors like 4A and 4B that can bind to your mRNA. So thus we have activated our mRNA. Now this activated mRNA is being guided to the 43S pre-initiation complex. We already have our 43S pre-initiation complex, is it not? So to this 43S pre-initiation complex, if this mRNA is going and attaching itself, then what will we get? We will get a 48S initiation complex. Alright, we get a 48S initiation complex, right? So now let us again draw this ribosomes translation machinery. First we have the 40S ribosome here, then we have a eukaryotic initiation factor 1, eukaryotic initiation factor 3, eukaryotic initiation factor 2 with GTP. Yes, and also we have a ternary complex, we have a tRNA with the methionine. Yes, and now our mRNA has come. Yes, now our mRNA has come. So the mRNA gets itself attached. So what guides this mRNA? The eukaryotic initiation factor 4F has guided this mRNA towards the ribosomal machinery. Alright, so now what is happening? The mRNA, how does this mRNA go and fix itself in the ribosomal complex? It fixes itself in such a way that the AUG codon has to be identified, is it not? The AUG codon has to be recognized by your ribosomal machinery, so that the tRNA that is carrying the methionine will be complementary, will have the anticodon complementary to that initiator codon. So for that, what happens, the ribosome will scan the mRNA to identify the initiator codon AUG. But how does it does this? How does this happen? So here in prokaryotes, what have we seen? We have seen something called as the Shine-Dalgarno sequence, is it not? Shine-Dalgarno sequence, that is in case of prokaryotes. So similar to that, in eukaryotes, we have something called as the Kozak consensus sequence. Okay, Kozak consensus sequence. So this Kozak consensus sequence, if it is identified, it is easy to identify the AUG codon, AUG first codon. Okay, initiator codon will be identified, and to that initiator codon that is situated 10 base pairs upstream of this Kozak consensus sequence, that will be considered as the initiator codon, and on that initiator codon the tRNA that is carrying the methionine will be placed, right? So this is nothing but your 48S pre-initiation complex. Yes, 48S initiation complex. Yes. So next, all things are ready now, is it not? We have the ribosomes, we have the first methionine tRNA in place, then you also have the mRNA in place, right? So what will be our next step? Our next step is to remove all the initiation factors out and to reassociate the 60S ribosome in place and form the 80S ribosome, is it not? So that is what we are going to do exactly. So what happens? We have another initiation factor, which is initiation factor 5. Yes, that is an ATP. Now initiation factor 5, that will hydrolyze your initiation factor 2 GTP. Yes, and converts it into GDP, right? And this is what happens, and this gets removed. Also other initiation factors like your initiation factor 1, initiation factor 3, initiation factor 4F, all of which gets removed. Yes, all of which gets removed. When all of these initiation factors get removed, then what will happen? Your ribosomes, the bigger ribosome, 60S ribosome is also going to come into place. It is going to reassociate with the 40S ribosome, and also you have your mRNA in place. Yes, we have the mRNA in place. So this mRNA is situated in such a way that your tRNA is, the methionine tRNA is in the peptidyl site. Yes, in which site is it present? It is in the peptidyl site. Also you have the acceptor site, which is vacant, which is ready to accept any new amino acid tRNA. Yes, so this is so as everything has reunited, what is this called? It is called as 80S initiation complex. Yes, 80S initiation complex. Now this is ready to enter into the process of elongation. So what is the process of elongation, all of you? The amino acid one by one, it is going to get attached, it is going to get linked with the previous amino acid with the peptide bond, and it is going to form a polypeptide chain. So that is what is the elongation process. And now we have prepared this ribosomes, mRNA and all that for the elongation process. Alright, so what are the components of 80S initiation complex, all of you? Here if you can see the components, you have the 40S and 60S reunited, you have the A site, P site which is present in the ribosome, you also have the methionine tRNA. Yes, methionine tRNA, which is the tRNA for the first codon, and the first codon AUG is in place. So this is present in your P site on the ribosome. Also you have your mRNA here. Alright, that's going to be read and translated now. So this is the 80S initiation complex. So all of the other initiation factors we have removed. right? So we had in prokaryotes also we have seen, if the initiation factors are these are removed and these will get recycled, is it not? But here in initiation factor 2, it's associated with GTP. So this again, how will it get recycled? With the help of 2B and 2C. Yes, eukaryotic initiation factor 2B and eukaryotic initiation factor 2C, with the help of this will get recycled back. Okay, after you recycle back, then again it will go and get attached to another fresh, again it will go to bind with another fresh amino acid tRNA. Yes, so this is what is the functions of all your eukaryotic initiation factors, right? So let us again just quickly revise what happens here. The first thing is, the first point is dissociation of our 80S ribosome. So 80S ribosome now will dissociate to 40S and 60S, and the ribosomes will have A and P sites only. Initiation factors 1 and 3, eukaryotic initiation factors 1 and 3 is going to bind with the 40S, and that will prevent in reassociation with the 60S, right? So also it will allow other eukaryotic initiation factors to bind to your 40S, because we have so much of eukaryotic initiation factors that have their function here, is it not? Yes. So next is formation of pre-initiation complex. So first a binary complex is formed with the help of the eukaryotic initiation factor 2 and GTP. Then when it binds with a methionine tRNA, that forms a ternary complex. Now this ternary complex, when it binds to your 40S, then that forms the 43S pre-initiation complex. And next is the formation of 48S initiation complex, in which you have to first activate your mRNA. So capping of the mRNA, to the cap what is attached? Eukaryotic initiation factor 4F is attached. So that is a complex which is made of 4A, 4G and 4E, right? And then also you have another protein, poly A tail binding protein, that will bind to tail. And also you have 4A and 4B proteins, that initiation factors, that will all of these will activate your mRNA. So this also needs ATP, right? And then this mRNA, activated mRNA, is guided to the 43S pre-initiation complex, and that forms your 48S initiation complex. Then the next step is the ribosome will be scanning this for the initiator codon, which is in the 5' AUG. How is it identified? It is identified by the help of Kozak consensus sequence, which is 10 base pairs upstream to your AUG codon, right? And then this 48S with this reassociated 60S ribosome will form the 80S initiation complex, right? And then what happens? You have to remove all the initiation factors. So eukaryotic initiation factors, factor 5 will hydrolyze your factor 2 with GTP, and it is recycled back with the help of 2B and 2C. And other eukaryotic initiation factors are also released, and they will be used up for the next cycle. Alright, so this is all about your initiation. Yes, this is all about your initiation. Yes, so next step is your elongation. Yes, the next step will be your elongation. So what is elongation, all of you? Elongation is nothing but addition of amino acids one by one by forming peptide bond and forming a polypeptide chain. That is going to be your elongation process. So again, this elongation process will involve 3 steps. So as told in prokaryotic translation, the 3 steps are the same, and it is going to happen in a cyclical process till the stop codon is being encountered. So the first step will be your binding of the amino acid tRNA. Yes, your binding of the new amino acid tRNA to your A site. Okay, to your A site. And then the second step is going to be your peptide bond formation. And the third step is going to be translocation. Yes, translocation. Now let's see the steps for one by one. The first thing is, let us now draw how our for 80S pre-initiation complex looked like. Okay, so we have the 40s ribosome, we have the 60s ribosome, and what are the different sites that you had? A site and the P site. Okay, A site and the P site, and you have your mRNA in place. Yes, you have your mRNA in the 5 prime 3 prime direction, and in the peptidyl site we had the AUG codon. So pertaining to this AUG codon, we had the methionine tRNA in the P site. Now our A site is vacant here. Now A site is vacant here. So what is this codon that is present in the A site? So that has to be first recognized. Yes, that has to be first recognized. So that corresponding to that codon, what is the amino acid? That amino acid is being taken up. Yes, that amino acid is being taken up, and the tRNA is charged with that amino acid. So let us say that is the amino acid 2. Okay, so to this amino acid 2 carrying tRNA, to this we have the eukaryotic elongation factor 1 that is also associated with GTP. Okay, that is associated with GTP. Now this guides this second amino acid to your acceptor site. Yes, to your acceptor site. Now as this happens, your now your tRNA is going to come and sit in the acceptor site. Okay, the tRNA that is carrying the second amino acid that is corresponding to the second codon is sitting in the acceptor site, right? So now here what will happen to the eukaryotic initiation factor 1 with GTP? That will again get recycled back. Okay, that will again get recycled back. So now it is going to get hydrolyzed. It is going to get hydrolyzed a similar way, like how we have seen all the other factors that is associated with GTP. It get hydrolyzed to GDP, and then with the help of other elongation factors, it will get recycled back. Okay. Now this now that we have two tRNAs with two amino acids. Now what is our purpose? We have to form the peptide bond between them, right? So how do we form the peptide bond here? Now the methionine is present in the P side and amino acid 2 is present in the A side. So how are we going to form the peptide bond? So the carboxy terminal of this methionine is going to link with the amino terminal of the second amino acid in the A site, right? So in this way the methionine is going to get linked with this amino acid. So this tRNA will break off this methionine. Yes, the tRNA will lose its methionine. tRNA in the P side that is going to lose its methionine. So that because methionine is going to form a peptide linkage with the second amino acid in the A site. So that is going to be the second step. So what is happening here? The peptide bond is being formed. So let us again draw it for a clearance. Yes, there is the 40S, there is the 60S ribosome. Then we have the acceptor site, then we have the peptidyl site. So the new amino acid has come to the acceptor site. So that is having the amino acid 2. Now in the peptidyl site we have the we have the methionine. So this methionine is going to get linked with the amino acid by a peptide, amino acid 2 by a peptide linkage, right? By a peptide linkage. So the N terminal of this methionine is free. The carboxy terminal of the methionine is linking with the N terminal of the amino acid 2. So thus the two peptide linkage is being formed here. Now this tRNA which is present in the P side is vacant. Sorry, it is empty. It is losing. It has lost its amino acid. Okay. So this is the peptide bond formation. Peptide bond formation. So what is the enzyme that is necessary for this? Peptidyl transferase. Yes, it is a peptidyl transferase. Peptidyl transferase. Now this step doesn't need energy. There is no energy required for this process. Why is it so? Because the amino acid is already activated. Is it not? Amino acid is already activated. While charging of tRNA, it is already activated. So in this peptide bond formation, you don't need energy. Alright. So this is how a peptide bond formation is formation has occurred. What is the next step? The next step is going to be the translocation. Yes, translocation. Now for the translocation to happen, let us again draw this. We have the 40S, we have the 60S, we have the mRNA, Yes, we have the mRNA, we have the A site, we have the P site, right? Now this mRNA is in the 5 prime 3 prime direction. Now what has happened here? The ribosome translocates to the next codon position. Yes, translocates to the next codon position. So here if I have, yes, if I have, again let me draw the previous picture. So I will be having the tRNA that has formed the peptide linkage like this, is it not? Methionine and amino acid, the two amino acids have formed the peptide linkage like this. So if my ribosome is going to move ahead to the next codon, then what will happen? Yes, then what will happen? The amino acid will move to the next codon. Then what will happen? Let me just move, sorry. Yes, it is just moved. So what is happening here? The A site is shifted to the next codon, is it not? The A site is shifted to the next codon. So it will be here, and the P site is shifted here. Yes, the P site will be here now, is it not? So the A site is shifted to the next codon, and P site is now shifted to the A site, that is carrying the tRNA with the polypeptide, growing polypeptide, right? So this A site has now become vacant. So this has become vacant so that a new amino acid tRNA that is carrying a third amino acid can come to the acceptor site. Yes, can come to the acceptor site. So what happens if it comes to the acceptor site? Now I have the third amino acid, and again a peptide linkage can happen between these two amino acids, amino acid 2 and amino acid 3. So again, what will be my picture? What will be my picture? I will have this kind of a, yes, I will have the methionine, amino acid 2 and amino acid 3, right? So this is how the fresh codon has happened. Yes, fresh codon has happened. Otherwise, what is the translocation step? Again, let us see. What is the translocation step, all of you? As this has happened, okay, as the ribosome has moved ahead to the to the next codon, the acceptor site has become vacant and peptidyl site is now carrying the growing polypeptide chain. So the A site has shifted to the P site here. Now in the P site, what did we have? In the P site, what did we have? We had the vacant tRNA, is it not? So there is no E site in this eukaryotes, whereas in prokaryotes we had an E site, but in eukaryotes there is no E site. Instead, the vacant tRNA will just be pushed off into the cytoplasm. Okay, vacant tRNA will just be pushed off into the cytoplasm. Alright, all of you. Yes, so thus we have just two sites, A site and P site. P site is now carrying the growing polypeptide and A site is now vacant to get the amino acid 3. Okay, so this amino acid 3 is like this. So this is all about translocation. So what step is this? It is translocation. This translocation is an energy requiring process. Yes, for this translocation you need energy. Alright, so let me again tell you how the third amino acid is going to get attached here. The third amino acid charged, the tRNA that is charged with the third amino acid will come to the acceptor site, right? So to the acceptor site, if the amino acid remains, then what will happen? The third amino acid will form peptide linkage with the second amino acid. Thus the growing polypeptide will now shift to that place, and then again the translocation process will take place and the ribosome will move one codon ahead. So the acceptor site will become empty again and the peptidyl site will be now carrying the growing polypeptide chain. So this kind of a cycle gets repeated. As this cycle gets repeated, the amino acids get added one by one and a new polypeptide is synthesized. Okay, and growing polypeptide is synthesized. Alright, all of you. So this is all about your elongation process. So let's just quickly revise. After the formation of initiation complex, the process starts. Elongation factors are being utilized, and there are 3 steps again: binding of incoming amino acid tRNA, peptide bond formation, and translocation gets repeated till the stop codon is encountered. So first, what do you have? You will have the methionine tRNA at the P site and A site is vacant. So that is in the ATS initiation complex. This is what you will have, right? And then the fresh amino acid tRNA will come to the A site. So what helps in this? Elongation factor 1 GTP will bring the new amino acid tRNA to the A site. After which this GDP is hydrolyzed and the elongation factor 1 is recycled back. And then the peptide bond formation happens. Here the C terminal of the methionine is linking with the N terminal of new amino acid by a peptide bond catalyzed by peptidyl transferase. It's a ribozyme. This step does not require energy because it is already activated in the activation phase, right? And then now in the P site the tRNA loses its amino acid as the peptide bond is formed. Now as it loses its amino acid, the tRNA will just exits the ribosome. It enters the cytoplasm. It is pushed off into the cytoplasm. Now in the translocation step, what happens? The ribosome translocates to the next codon towards the 3' end of the mRNA. Now what helps in this process? Elongation factor 2 with GTP helps in this process. So it's an energy requiring process, right? So now what has the ribosome translocates, the tRNA with the polypeptide chain will move from the A site to P site. So please imagine this. Yes, go back, draw again and again. Yes, as I had drawn, please draw it in your notebook so that you understand it better, right? So as ribosome translocates to the next codon, the chain also shifts from A site, it shifts to P site. So always remember, peptidyl site will have the growing polypeptide chain. Acceptor site should be made empty so that it accepts the new amino acid tRNA. Alright, now empty tRNA is pushed out, right? So this is the process of elongation. This process repeats till the stop codon is encountered. So hope you understood this elongation phase. So let's move on to the termination phase. Right, so in termination phase, what are the stop codons? UAA, UAG, and UGA. When the acceptor site recognizes the stop codons, the release factors, eukaryotic release factors come into play. They will bind and stop the process. They also break the bond between your tRNA and the growing polypeptide, and it will release them. So this process also needs energy. And now also these release factors will dissociate the ATS ribosomes to 40S and 60S, and it will prepare them for the next cycle of translation. Alright, so this is what happens in the initiation, elongation, termination. Alright, so please go through again and again. It's a very easy process. So just remember the factors. Okay, just remember the factors, by heart the factors, and the process is very easy if you learn it in steps, right? So energetics, if you see, it is again 4 high energy phosphates we have used. 2 high energy phosphates we have used to charge the tRNA with the amino acid, and 1 high energy phosphate we have used to bring the amino acid tRNA to the ribosome, and 1 more in the process of translocation. So this this 4 high energy phosphates that I am mentioning, it is to attach each amino acid to the polypeptide chain. Okay, so to attach every amino acid to the polypeptide chain, you will need 4 high energy phosphates. In addition to that, you know, in general you will be using ATP to activate your mRNA, yes, for termination process. So in all these you use, in general you use the energy, but for attachment of every amino acid to the polypeptide chain, you will be using only 4 high energy phosphates. Alright, so this is all about your translation in eukaryotes. Alright, yes.